Are you seeing it now?
Yeah, we can see. Yeah, it's all good.
Wonderful. Okay. So anyway, as Tony's saying, so over the years we've developed a number of robots, all biologically inspired, and that's what some of the things you're seeing here. So we've been successful in doing a number of things in terms of both creating robots that do interesting things, but also modeling the biology, which is just as important to us. So this is a slide I like to show. We learn biological principles from animals. We apply the principles to improve robot mobility and autonomy, and we use the robots we develop as models to test biological hypotheses. So it's not just about the robots, it's also about the biology. And this can't be done without biologists, of course. And here are three fantastic biologists I've had the good fortune to work with in my career at Case, Hillel Cheel, Roy Ritzman, and Mark Willis. And of course, there's a number outside of Case, many outside of Case that we've worked with over the years. So this outline, I'm going to talk a brief look at our 30-year history of this group, and then talk about synthetic nervous systems a bit, and then neuromechanical scaling, and then if there's time, some fun with Whegs. So going way back, we started Randy Beer, Hillel Cheel, Leon Sterling back in 1980, had this artificial insect project, which was simulation. It was all simulation, and it was a cool neural network that had not existed before. The concept was completely different, and it was based on what was then known about how insects worked. And I was brought in, the question is, can you make a legged robot? Can you control a legged robot without neural simulation? And of course, the answer is yes, and you see it there. So then Roy Ritzman got involved because he was a cockroach expert on campus. So the question is, how do you build a cockroach robot? Well, what's the mechanics look like? Nobody really knew at that time what's cockroach mechanics look like, how the front legs, middle legs, rear legs work, how all the joints work. Recording EMGs at the same time. So we can figure out that the front legs are more like arms, and so we modeled this with 5 joints in the front leg. The middle legs are a little less complex, so we modeled with 4. The rear legs are more like pistons to drive the animal forward. Here you see active control, and here's Gabe Nelson over here, and he's beating on the robot, showing that with active control it's stabilized. Gabe went on to control Big Dog and all the Boston Dynamics robots through Atlas before he recently left. So that was with active control, but if you use artificial muscles like this, you can get the same kind of stability with just passive mechanisms, so that the muscles have the passive stiffness. So this is, of course, well known in okay, so then something else we built is this cricket robot, which is all 3 inches long. It also uses these McKibben artificial muscles, which we had to build. They're only like 1 inch long. And of course, McKibbens are pneumatic. So that's where all these hoses are in the back here, driving it. So now, those robots you just saw were controlled with finite state machines. So the whole idea was to mimic the behavior of the animals we were modeling. But there wasn't enough known about the neuroscience to really use the neuroscience directly. But 20 years, there was like 20 years of research since we this work with neural networks. And during that time, a whole lot more has been discovered about neuroscience in these animals. So the question is, could we actually use computational neuroscience approaches to develop what we call synthetic nervous systems, which are biologically plausible dynamic neural networks with the form and function of the animal nervous system? So the question is, could we do that? And this is something that this paper got us very excited. It was from 2004. It was a modeling paper, but the modeling paper actually, of course, modeled data from many years. This is Ansgar Busch's from Cologne. And it's actually a very detailed model, and it shows sensory connections. It shows that there's they know there's a central pattern generator each joint of an insect leg. And then the sensory feedback coordinates the joints in the leg. So all this was known. And this guy was very excited. And then also in 2010, David Coffer et al. Published this paper on AnimatLab, which is a software that you can build a neural system and a mechanical system in the same software, and they run together in parallel. So this is a really wonderful tool. It's free. So we found this about the same time. And insect nervous system, of course, there's a brain. We call it the central part of the brain is called the central complex. There's a nasal ganglion, which is like a brain stem in a vertebrate, and then the ventral nerve cord, which is like a spinal cord in a vertebrate. So there's quite a bit known, not as much as we like, of course, but there's quite a bit known. So we thought, okay, why not design a synthetic nervous system based on what is known? And these are not your regular artificial networks. These are each neuron is modeled by a first order ordinary differential equation, and they talk to each other by way of synaptic connections. And you can see here the presynaptic.
Did we lose Roger?
Looks like it.
Yes.
Okay. Oh, that's a shame. Well,
Hopefully come back.
I have his phone number if we need to call him.
yeah, go hassle him. It's a great excuse just to make his life even more difficult. But as we wait for Roger, Nathan, shall we follow up on your question? Because for me, the challenge we face is how do we make progress in this agenda? Because I feel very often, because we want to make things testable and concrete, also Holger, it would be good to hear your view on that. And Tony, we again start to fall in these reductionist traps. And we'll leave lots of things outside of the discussion because we have to make it comprehensible and testable. So how can we make progress on this more integrative agenda?
Is that a question for me?
Well, you're a smart guy, right? So yeah.
So my slides later, I was kind of touch on this a bit to provoke some discussion.
Your living machine poster behind you is not straight. That's very disruptive.
Yeah, I know it needs to be sorted out.
Okay,
but,
good.
So I guess my view on this would be, I mean, deep learning is becoming the main paradigm for neuroscience now, arguably, whether that's the right way or the wrong way for the field to go. It does seem to be the way the field is going. So I guess there's a question there, how you combine that flow of the field with, all these insights you've given on and the history of living machines and paradigms for human intelligence.
Yeah. So I don't think, well, I don't have an issue with machine learning and artificial intelligence, but it is as a method for us to analyze data, say, I think it is very useful. There's no question about that. But conceptually, I'm not entirely sure that we will learn a lot from the new AI trends. But we can get back to that because Roger is back. Sorry.
Yeah, sorry about that. This is the dangers of. There we go. I think I'm back. Anyway, okay, so I was talking about the CPG we use, and the point is that there's a tonic drive that excites the half centers, and with no tonic drive, there's
Not
no
for your
oscillation.
slides, Roger.
What happened? Oh, darn it.
Maybe the
What
others
happened?
can stop the video.
Nothing,
Are we okay?
nothing. Yeah, nothing against your appearance, Roger. Okay, don't take it personal. Just the slides.
Is it? Are we okay, or do I have to change something?
No, we still can't see your slides.
Darn it. Oh, okay. I know what I did wrong. Darn it.
Is that your
Excited.
biomimetic user interface?
Yeah. Oh boy.
That's
All
it.
right.
Great, we got it.
Yeah. Darn it. All right. Okay, hopefully we're there now, right?
Yeah.
Okay, so anyway, the bottom line, this is oscillation frequency versus tonic drive, and with increasing tonic drive, you have a higher, a faster oscillation in the CPG, which of course is what you want. Now we have what we call a functional subnetwork approach to design synthetic nervous systems. And the idea is the neurobiologists perform recordings that elucidate nervous system structure. We mimic the structure and then tune functional subnetworks directly by exploiting neural dynamics. The resulting subnetworks can be assembled into larger hierarchical networks that function as intended with no additional tuning. So the idea is we don't have to do naive optimization after the fact. We can build and tune as we go, and we have a functioning network. So all parameters values can be input from neuroscience data or selected directly based on the network architecture. So this is a functional step network approach. And what's really awesome is you can add subnetworks and functions without the need for retuning the entire system. So I love showing this slide. This is the first successful synthetic nervous system. Nick did this. And this is for one leg of a cockroach. And here you see 1, 2, 3, 4, 5 CPGs. That means there's 5 joints in this leg. And now there you go. It's all 6 legs. Every leg has one of these controllers. And then of course you need interleg pathways, also from the literature as much as possible. And the result is our very encouraging simulation, cockroach walking. And if you look at the animal data versus the model data, you look at these are different joints in the middle leg, the same joints in the same middle leg. And you see the arrows show you that there's agreement in trends. And if you, by modulating muscle control, you can actually change the turning radius as this simulation is walking. So we were very encouraged by this. Even Roy Ritzman liked it. So that says a lot. He's very particular when it comes to especially neuroscience. So 3 years later, one of our students, Scott Rubio, basically improved the biomechanics of that model, improved the interleg coordinations, and we have a much, much nicer simulation. It looks very cockroach-like. And again, praise from Roy Ritzman, which is, like I say, that's what we're going for here. So, all right, so getting back to the big picture of the nervous system, insect's nervous system, you got the central complex up here, the gateway ganglion here, it's like the brainstem, and then the ventral nerve cord. And then there are descending commands through the nectar connectors, but they're also ascending sensory information. And we're very interested in, and just one information is being transmitted back and forth between just these neck connectors. And the things that we know is that the brain is not a micromanager. We know that the local circuit is doing most of the work. Well, you also know that the information is very important coming from the brain for volitional movements. So this is Roy's lab, and here you see he's actually got microelectrodes into the insect brain, in the cockroach brain here. He's recording from the central complex as the animal's behaving. And for example, here's a unit that's encoded to left turns. And here's a unit that's encoded to right turns, faster right turns. And here's a unit that's encoded to left or right turns. And so Nick built this praying mantis, and it's tracking this light. So as you move this light around, you see the head, the neck is rotating. There's a body joint also rotating. It's got one leg doing the correct motions such that if it could, it would be following and tracking that light like prey. So this is a case where they're sending information from these light sensors in the head are being used for commanding motion. So we also do work in rats, and this is Martin Fischer at the University of Vienna, and this is a rat on a treadmill, and there's holes, there's a hole, and that leg right there just fell in the hole. The rat can't see the hole, so it has to react to it. So it's a perturbation, and what you find from the data is that there's an expectation of touchdown, which indicates CPGs. And there's also a very rapid reaction to perturbations, this perturbation, like 5 milliseconds, there's a reaction that you can see in the EMG data. No. So Alex Hunt modeled a synthetic nervous system for the rat. And this is for a single leg. And what you see here is it's a simplified system, simplified graphic here. But there's a hip, knee, and ankle half centers for the CPGs for each of those joints. And then there's ear neurons and sensory in the sensors. And what's important to note here is that down here you see there's a color coding. So purple is from Pearson 2008. These connections are all from, blue is from Prohaska, brown is from McBee. And there's just a couple here in black that Alex had to figure out on his own to make the system work. Doesn't mean it's there, doesn't mean it's not there, but it does mean that it's certainly hypothetical, something to look for. And that was for one leg, and then here's for the 4 legs. Here's a left front, right hind, left hind. And there's connections here as well, the interleg coordinations. And that's from data from cats and mice. And again, the green is from Mackay and the gold is from Talipar. And there's just a connection here and here, it's in black. You can see that Alex had to figure out. And here's the rat walking, and you'll see it starting, it's uncoordinated, now it's coordinated walking. And then he's going to perturb it. And when it's perturbed, it will get out of coordination, but then get back into coordination. So also, this is also very encouraging. And then what Alex found is that there's 2 sets of data, 2 sets of rat data in terms of gait. And what he found is there's just, with changing just one synaptic conductance, he could either get this gait or this gait. Also very encouraging. And then Caillou Deng, who's working with Alex and me. Alex is now at Portland State, just tenured. Congratulations to Alex. So Caillou Deng's working is a student that we're working with. And what Caillou managed to do is reproduce what's called resetting deletion in rat experiments. What it means is, over here you see hip, knee, and ankle, and you see just nice coordinated movement. And there's a perturbation, so it comes out of coordination. But then after some time, the perturbation goes away, and we have back to the original coordination. So this is called a resetting. That's what it's called, resetting, because the gait resets despite the perturbation. So Caillou managed to reproduce this with a synthetic nervous system. Alex's synthetic nervous system, and actually make it a two-layer structure with a rhythm generator network on top of the CPG, and below that this pattern formation networks, and each in the pattern formation network includes CPGs at every joint. And this is hypothetical, but somewhat accepted, fairly accepted out there in the computational neuroscience world. And this is the dog, the Greyhound robot that Alex controlled with a synthetic nervous system. We took the rat system and he scaled it up for the dog. So now I want to talk about scaling. This is very important to us because it's really hard to build robots, for example, the size of a fruit fly. That's one reason we want to do it. The other reason is we just want to understand animals better. So this is Greg Sutton at the University of Lincoln, Nick Jasinski now at West Virginia University, Phil Chill at Case. So is it humanistic insects joints, and you can a simple model of a joint. So here's a pivot and here's a muscle, extensor and a flexor muscle, and they're modeled with stiffness and damping, and also an active contractile element. And of course there's gravity, so it acts like a pendulum. In swing, here we're modeling swing. So in swing is, and so we have for because it's a pendulum, we have a positive stiffness from gravity. There's of course elastic elements from the spring from the muscles and tendons, and then there's viscous effects, and then there's inertia. So here's the equation of motion, just nice simple linear system theory. So if we're modeling swing, and swing is so an unloaded leg swinging in the air, just back and forth, cycling. So if we model that as a harmonic, as a cosine, then the solution to that is this linear system theory. So this is just also a cosine, but with a phase lag. And that phase lag depends upon damping, stiffness, and inertia, and also the frequency of the isolation. So this phi is the phase lag that you see. It's the difference, it's the phase lag between the motion and the torque. Okay, so if we look at this speedometer, this is basically a phase angle speedometer from 0 degrees over to 180 degrees. So if you look at this equation and you just plug in values, you can plug in if C is 0 and J is 0, you end up with a phase angle of 0. So for elastic effects, you've got a 0 phase angle. But if you plug in C is not 0 and these other terms are 0, then you end up with 90 degrees phase angle. And then, so the viscous effects give us a 90 degrees phase angle. And then if you plug in C is 0 and K is 0, and just leave J in there, you find because of that minus sign, you have 180 degrees phase. So inertia gives you 180 degrees, viscous gives you 90 degrees, and springs give you 0. So for this particular example, the phase angle is like 150 degrees. And when you combine these up vectorially like this, you get a 150 degree phase angle. So, okay, but the point here is that the phase angle depends on C, K, it depends on the system parameters and the speed. But the system parameters scale with length. We know mass scales with length cubed. But we can do better than that because our limbs, our animal limbs and animal bodies are basically tubes filled with water. So given that fact that they're basically cylindrical and filled with water, from that you can actually come up with a constant of about 10, and it actually works for very small animals all the way up to whales. And this was done, this McMahon.
Unfortunately, I think we lost Roger again.
Oh,
There he's coming back.
I'm starting to think that Zoom hates my talk. So I'm going to get us back here. I'm sorry.
Maybe you could also shut off your video. Maybe then the bandwidth will be better.
I will try that. No one wants to see me. All right. I'm going to turn off my video. That's an excellent plan. Okay, so now I'm going to share the screen and share. And now slideshow. Darn it. Okay, slideshow. I think I'd be getting good at this by now. Come on. There we go. Now.
We can't see anything right now.
Like I said, you think I'd be getting good at this, but you'd be wrong. All right. Share the screen. There we go. Darn it. Slideshow. From the current slide. Wonderful. Now display.
It's perfect. Now we see everything.
Thank you. Okay. All right. So the point is we can scale, all these parameters scale with length like this. And then the rotary analog, of course, you know, it's a joint, it's rotating. So we can, that's a simple transformation from these linear scales to the rotary scales. So, and remember that the inertia it has a phase angle of 180 degrees. The viscous terms give us a phase angle of 90 degrees. The elastic and gravity give us 0 degrees. All right, so we can plot then the phase angle. Now that everything's in terms of length and time, we could use speed, but instead we chose to use time, and time is just the inverse of speed. And time, of course, in this case is the cycle time, the period of motion of this leg that's swinging. So we can plot then the phase angle versus length and time. Well, we could do that three-dimensionally. We use a heat map. And phi is color-coded in this. So yellow for kinetic, orange for viscous, and red for quasi-static. And here's what it looks like. This is length versus time. And what you see up here is a kinetic region. This is the inertia-dominated region. And then you see down here, the orange region is the viscously dominated region. And then the called quasi-static region. So what does this tell us? Well, what's interesting is that this is the resonant frequency line. There's critical damping here, which means that above that, it's underdamped. Up here, there's less damping. The higher you go this way in length, the less damping you find. And then below that line, below the dashed line, it's overdamped. In other words, if you pluck it, it doesn't vibrate. So you only have vibrations above the dashed line here. All right. So the idea is that the actuator is putting work into the system, And if you're operating over here, it's the work is going into kinetic energy, creating kinetic energy. If you're over here, the work is stretching springs essentially. And over here, the work is being dissipated by the viscous damping. Okay, you can break this up into 5 regions, the region 1, 2, 3, 4, 5. And what you will find is that, well, something I want to point out before we go further is that remember, this is phase plotted as a heat map. Up here, with this underdamped, notice it's a continuum. So you see the blurring of orange and red. But up here, you see a very clean line. That's because when it's really hot, when there's very little damping, you can go from 0 degrees to 180 degrees very rapidly. We're down here with this lot of damping, you're actually going from 0 degrees in the red through 90 degrees in the viscous up to 180 degrees here. So it's actually a very slow, very gradual change. All right, so you can project behaviors onto this map from animals. You can also estimate failure stresses, and that's where, so now these are animals dropped onto this chart. But before I talk about that, look at this line here. This is the bone ultimate stress line. So you don't see any animals over here because they would break their bones. Also, this is the muscle ultimate stress line here. You also, of course, you don't see animals beyond that line because, again, they would tear the muscles. So this is just dropping animals onto this plot. And you see way down here, the fruit fly is operating the viscous region. And then you see the mite also viscous region. So you see all these insects. The American cockroach is actually moving between the quasi-static and depending how fast it's going, between quasi-static and viscous. And you see the rat here is starting to approach this interesting point here. The rat is in this, this interesting transition region. But the cat is up here in the operating along the line between quasi-static and kinetic. And dogs are, of course, right up here also. Humans were in the kinetic region. Elephants also up in this region. Emu, notice it's dangerously close to the ultimate stress. But this is interesting. But remember, that was only for cyclic unloaded movement. That was not for stance. So I just repeated that same plot right here. So this is just swing. If you do the same analysis for stance, now the big difference is in stance, legs have all that body mass that they're supporting. So now the mass goes way up during stance. And that's why the kinetic region, which is over here, now drops into this much larger region. So for example, whereas the cat over here was just, oh, by the way, the other thing I should point out is on the last plot, you saw lines over here. Here we're showing just points because that's more accurate. In swing, the speed of swing is relatively constant for animals. The speed of the swing phase is relatively constant. So here we're just showing it as points, which makes more sense. Over here, stance, yes, stance, that's where the speed changes. So slow walking to fast walking to running. So it's interesting. The fruit fly is still down here in that viscous region. The American cockroach is still sort of spanning, but all these other animals in stance are more kinetic. Okay, so the point is the phase angle phi is a dimensionless number for comparing motions. Phi depends, so given MC and K, they depend on length, yeah, but given MC and K, giving speed or the cycle time, you have a phase angle. So this dimensionless number, phi is measured in radians, I showed it in degrees, but of course you can measure in radians. So it's a dimensionless number that includes gravitational acceleration, elastic and viscous forces. It enables us to compare virtually any two motions. So let's look at examples. So here's the horse. The horse has horse leg. This is swing. We were talking about swing in particular here. So here are the numbers for a horse. And you see the mass is significant, and the stiffness is the stiffening terms are significant. And you put that together, there's very little damping. So you put that together and you end up with like a phase angle like 178 degrees. So sure enough, the applied force and displacement are almost 180 degrees out of phase. So it's very much a kinetic beast. If you instead look at the stick insect, you look at the numbers for the stick insect, and you see very small mass, a lot of damping. And because of that, we have the stiffness part plus the damping, and we end with a phase angle about 17 degrees. And sure enough, that means that the applied force and displacement are roughly in phase. Now, here's what I call the horsey stick insect, and everything stays the same except if you speed up the motion, enough, you can make the stick insect have the same phase angle as the horse, or roughly the same. So with this ridiculously fast cycle rate, we see a phase angle of like 173 degrees, which is similar to the horse. So, and now it's out of phase. Of course, stick insects don't do this, but this tells you that you can do this when you're building robots and you're trying to scale robots. Okay, so summarizing the scaling argument, the idea is that to conduct cross-scale, cross-phylum comparison motion, we developed a dimensionless number that considers a balance of many forces, and that's the phase angle phi. So I talked about I showed the 5 regions of motion, on that plot. And I didn't talk about them specifically, but there are 5 regions, and animals exist in most of those regions. You can find animals that exist in most of those regions. The question is, are there going to be different neural solutions for the different regions? And of course, we think there will be. Now, dynamic scaling of robots. Well, with robots, you can artificially put in springs anywhere you want. The motors already have damping. So what you can do is you can take this stick insect robot, which is 100 times larger. I'm sorry, I said stick insect. This fruit fly robot, which is 100 times larger than the fruit fly, but you can purposely design it such that it has the same value of phi. So it actually, even though it's 100 times larger, it's dynamically at scale to be similar to the little tiny fruit fly. So, as I said, we expect the animal size and speed to strongly affect neural control mechanisms. This is what we expect. We're picking on rats and dogs, rat here and the dogs up here. Sea slug, which is the sea slug feeding, is also a cyclic behavior, and it goes between loaded and unloaded behavior, and it's quasi-static, so it's well into the red, where the fruit fly is well into the orange. So we have chosen these different animals to study because we want to understand the differences in the nervous system in these different regions. Okay, so this is just a repeat of the same plots. And so the question is, can an animal size speed scale be used to effectively predict the structure of low-level motor control networks that produce muscle activation? That's the question. So here's Fletcher Young, and he's built this very complex model of a rat hind limb. It's got 3D muscles, and the idea is to study the muscle activation properties of an entire limb. So what I showed you before was a very simple model, linear system theory, one limb. Here we have multiple segments and many muscles. So what, of course, the muscles he's modeling as a linear hill muscle model. And This is being done in AniMATLab. Includes passive and active components. So here's the derivative of tension with respect to time of a muscle. Okay, so so what Fletcher has done is he's developed custom optimization routines so that he can figure out what the torques and EMG should look like for behaviors of a rat, hind leg. And he can compare that with actual EMGs to tune it properly. Okay, so then what he did, now we had the rat working properly, the rat hind leg working properly. Then what he did was he just scaled the rat up. So up and down. So here's a very small rat and here's a very large rat. Without changing anything else, he just changed the length. And as he changed the length, of course, the mass properties, stiffness, damping, everything changes. So here's one example of what happens with a knee extensor. So for a large animal, we're up here, this is a large, very, very large rat, it would be very scary. The extensor is bursting in early to mid swing. And when it's small, on the other hand, for small, the smaller sized rats, it bursts in later stages. So the point is that just by changing the scale, we see a change in activation of muscles. And this tells us, of course, a change in the nervous system that's making it happen. Okay, so to better understand insect neuromechanics, we construct robots in the loop with computational neuroscience models. That's what you're seeing here. What you see here is a robot that's actually in the loop with this synthetic nervous system. When I say in the loop, that means that the outputs of this synthetic nervous system include motor commands to these motors, which are driving this robot. There's sensors on the robot, position sensors and strain sensors that get fed back in. So this is a robot in the loop running real time. So, so we can investigate specific questions. For example, the observation that each time a stick insect leg takes a step, sensory feedback from the leg excites a descending neuron that further activates walking behavior. And this is from a paper by Stoltz et al. To understand this data fits in the bigger picture of the nervous system, we integrate this result from numerous other studies. I say numerous other studies. Clarissa Goldsmith here, working with Nick at West Virginia University, they scour the literature looking for every bit of neuroscience, insect neuroscience that they can use to fill in the blanks here. And where they can't fill in the blank, or where they can't find the data, then they use a functional subnetwork approach for further development and tuning. The bottom line here is that we don't want to build, I heard Paul earlier say, you can use AI and you can make this opaque network, but it's, it creates the behavior you want, but it's just opaque. You don't know, you haven't learned anything about neuroscience. So we want to learn the neuroscience as well as control robots. So the bottom line here is that we successfully did in fact closely match the behavior that has been measured in insect by insect neuroscientists. Okay, so we've done two things. I'm repeating myself, I can't help it. We've done two. Yes.
We're running out of time here. Because we lost.
Okay.
Have you got much left?
Okay, I'm almost done. So we're doing two things. We're learning about the neuroscience, and we're also learning how to better control these robots. Okay, so this one, researchers at Carnegie Mellon, University of Lincoln, and are working on understanding communication, coordination, and control. Darn it. There we go. And this is a sea slug. So they, Hillel is a neuroscientist here, recording from mussels. EMGs from muscles and also recording from the nervous system, and then developing biomechanical models, and then developing controllers that match the behaviors. But reinforcement learning, again, you know, you can match the behaviors, but you don't really understand much about the biology. There's a Boolean network that they recently published that actually does explain some of the biology, but we're now working on developing a synthetic nervous system that is very much informed by the nervous system. Okay, and of course, building robots so I don't get to talk about funds with Whegs, but I'm just going to show, here we go. This is the whole idea is how can you make a Whegs robot run really fast and still climb? And the answer is put the 3-speed transmission in it. And here it is climbing over stuff. And it shifts and it takes off and runs at 25 miles an hour. So this is outrunning a human. And this is, here it is running at 25 miles an hour. So it's fastest legged robot in quotes. Do I have just a couple more minutes, Tony, or should I just stop? I can stop if you want.
I think if we carry on much longer, we'll miss our coffee break.
Oh, we don't want to miss coffee. Okay. All right. I'll just stop right there.
Okay.
Okay.
Well, thank you very much, Roger. And I think for the interest of time, we will also leave the questions for Discord. So please post questions in Discord for Roger. Roger, are you able to get onto Discord?
I haven't tried yet. I'm sorry.
Okay, you can also post questions in the chat here.